All right, well, thanks. It's great to be here. This is indeed loud. So we actually did buy a T7, and I'll tell you about our experience working with Complete Genomics and using the T7. And specifically, I'll mostly talk about these first two topics, and then I'll just say we are using the first two, by the way, are genome sequencing. I want to actually talk a little bit more about RNA sequencing with their technology. We'll talk about spatial, where we're doing quite a bit of spatial in the lab these days, and then just to mention that we are doing Olink's, the sequencing readout with Complete Genomics technology as well. So many of you may know that Complete Genomics has a nanoball kind of technology for getting very high density nanoballs and sequencing, and they've also added on this long fragment read technology that basically adds barcodes and lets you link up variants. And so about 2 years ago, before we got our sequencer, we actually tried this technology in collaboration with them. And basically, of course, we sequenced my genome, which has been sequenced many, many times by now, including Hassan, who's here. So we sequenced it. It's become a bit of reference for the lab. We sequenced with straight up short reads for 40x coverage, and then we did do this long fragment read technology for 30x coverage. And it actually did seem to perform quite well. We got the typical number, in fact, even better than we got for Illumina, 3.8 million variants of very high confidence, and we've done a lot of QC on this, and we think the technology is roughly equivalent to Illumina. It actually, the genome came out better than we'd done for Illumina, but in fairness, we had sequenced deeper. So I think the accuracy is about similar for the SNPs and indels. And then we called variants, and you can see the variants by Illumina, again, variants by Complete Genomics. You get pretty much the same distribution of types of variants, and again, the overlap was quite high. I won't show all that. The nice thing was with this long fragment read technology, you really could link up all the variants. And so it really did lead to a nice phased genome with this extra, which winds up being about twice the cost. They can comment if I've got that wrong. But in the end, we wound up with a pretty well phased genome, and it went all quite rapidly. So I think that was quite nice. Now, more recently, they've come up with a similar procedure that uses long fragment reads for RNA so that you can actually map out isoforms with RNA. And I won't go through all the details of this. It's a slightly different protocol. You use uracil removal and such. But the net result is the same. You wind up inserting these barcodes into basically the same fragment, if you will, so that you can essentially link all the reads from the same fragment with the same barcode, and then at the end know which goes with what. And you'll see why that's important later for allele-specific expression. And so we did build a pipeline for this. I think they have their own, but I'm not sure if it was ready at the time. But Jennifer Lee-Pouton did this and built a pipeline for quantifying RNA levels, also for calling allele-specific expression, for calling editing. That's fairly new. I'm not sure that's on the slide yet. And then, in the end, we can link up all the barcodes and get better allele-specific calling, you'll see in a minute. So basically, again, you can just do straight up RNA sequencing and get your genes and call pathways like this. I happen to be diabetic, and you can pick that up in our RNA-seq. This is from PBMCs, I forgot to say. And here's the part that we're getting more and more interested in, this allele-specific expression. And there is a lot of this in the genome. So, if things are expressed identically, each allele would be expressed 50%. So the number would be 0.5, but there is quite a bit of a distribution. You can see here with different alleles getting expressed at different levels. And as a general rule, reference alleles tend to be expressed higher than the minor allele. Well, I should watch that. The major allele gets expressed higher than the minor allele in general, but there are many exceptions to that. It's very individual specific. And we actually, I believe, were able to call more than we were done with a project we had done in 2012. And these alleles, they actually do have important information, and they'll have missense substitutions, they will have frame shifts and other things. And so you can actually pick all that up in the RNA-seq. And there's a fair amount of that that does go on. Now, the nice result is here with these linked reads, you can actually, every sequence, as long as it's linked with its barcode, you know what isoform it goes to. So therefore, you can actually, for those of you who've done allele-specific expression, when you try and just call it with the variant itself, you don't get that many reads across the variant, right? And that's a problem. So you don't get that, you can only really call it for the most abundant genes when you get enough reads. But when you have these linked barcodes, you're able to get every read counts. And so therefore you get much, much better quantification. And so you can get down to very low level expressed genes and so get allele specific expression. Does that make sense? And that's the power of having all these linked reads. And so in the end, we could basically call that. So meaning if you have a barcode off a read over here and you know it's linked to that allele versus a small allele, you can make all those calls. And so the net result then is you can start making these comparisons. And this is analysis in progress. We're very interested in how does allele-specific expression change over time. And some of you may know we've been running a study on roughly 100 people for now 11 and a half years. And so we're trying to see how their allele-specific expression might change over time. And this one happens to be me. This is my 2012 data. Basically, this allele was expressed at about 0.4 compared to the other one at 0.6. And we call these flippers. In 2024, the relative expression flipped. So you can actually quantify this, and we think that we'll have to see what the biological significance of all this is, but some of these genes are pretty interesting. And so, excuse me, that's the plan. We're now doing this more systematically to call all of this. The other way that we're working a lot with complete genomics is with spatial. As I mentioned, we're doing a lot of spatial these days of all kinds of targeted technologies. And what's special about complete genomics is they have a platform called Stereo-seq. There's been, excuse me, several publications out on this. And it's a sequencing-based readout rather than a targeted readout. So I don't know, I wasn't going to explain this in a lot of detail, but how many of you know about spatial technologies? Most here do, right? Good, all right. So basically for most technologies, you have defined probes that have all been worked out. You have to optimize them. And then you detect typically, well, this one does about 100 probes. Might be a little low. There it is. And then, you know, when they first were rolling out, CosmX had 1,000, Xenium 480, Merfish about 500. Now they've scaled somewhat higher, although the price has gone up quite a bit. So with Stereo-seq, you can actually, I'll show you in a minute, you do a sequencing-based readout. Now they do have different field sizes, and that's significant because that affects your price a lot. So don't just worry about what the price per slide is, know how big your slide is, because a slide that's twice as big will get you, half the cost because you load it up with as many sections as you can. So anyway, these things have all been worked out quite a bit. And Chen in our lab has actually really been spearheading a platform comparison amongst these. And we have our favorite targeted assay, and then we do use Stereo-seq for untargeted. They give complementary information. So I won't get too deep into the details on this either, but basically you have a pattern, if you will, chip that you have essentially barcodes down on. And on this chip, you have primers, if you will, and then you lay your tissue section on top of that, and then you prime and are able to sequence or do primer extension, and then you loot everything off and you sequence. And because you know the position of the pattern cell, you can actually relay back in a spatial fashion where the different sequences came from. And what's nice is that the spot size on these is quite high resolution, 220 these nanoballs, 220 nanometers, although you don't get full information from one nanoball. Don't let that fool you. You actually have to do a bit of a sliding window where you're integrating over multiple, if you will, pixels of these nanoballs to be able to get your information. And it does work quite well. In the end, you wind up getting, well, I'll show you more later, but about 250 transcripts. That's total transcripts per cell. Now that sounds kind of miserly, but that's actually pretty good for this kind of analysis. And, for this one first section, some of the first experiments we did, we sequenced quite deeply, 4 billion reads, which is now possible because this stuff is getting fairly cheap. This is over about 200,000 cells. And in the end, the nice thing is you can really cover a lot of gene space and pick up a lot of transcripts. So the data gets very, very rich very fast. And what you'll see, we're doing this for multiple regions of the intestine, both small and large. This happens to be a region of the small intestine nearest your stomach. And you can see you can pick up many, many different cell types because there's enough transcript information to define what each cell type is. I'll go through some of these numbers in a minute. And of course, it makes beautiful pictures as well, but you can drill in and see all the different genes. And we and others are working on trying to identify splicing isoforms, which is kind of nice from the sequencing technologies, which you won't necessarily get from targeted assays unless you've designed probes that way. So anyway, it works well. That was this one region, small intestine. Here are other regions as well. Again, you can map out all the cell types, and we can assign nearly all cells. There's some work to get the segmentation right. We've also been doing a lot to study this cancer. It's a colon cancer model, if you will. It's called familial adenomatosis polyposis. These folks have an APC mutation, which is thought to be the initiating event in colon cancer. And so what happens is these folks with 100% frequency will get colon cancer, and they get hundreds, if not thousands, of polyps in their colon. And so basically, it's thought that these polyps are a model for early stage cancer. They're not yet cancerous, although occasionally an adenocarcinoma will pop up. And so you can take these polyps of different sizes, different forms of dysplasia, and actually sequence them. And so we've been doing that and have run Stereo-seq on these various polyps, and it does work quite well. Once again, from deep sequencing, you can, in this case, even more when you sum over all the polyps we've done, you can start defining all the cell types that are there and look at, cancer-associated fibroblasts, all the different things, and try and map out stages. That's a whole separate project. So now into some of the technical performance of this. The replicates are quite good. So replicate experiments have 0.98 correlation. If we compare, nice thing about this one, you can do it on flash OCT as well as formaldehyde fixed. And the correlation's pretty good. Things are stored a little bit when you have OCT. As you may know, the fixed sections are really quite stable and good. One of the nice things, and this makes sense, with the sequencing-based readout, it actually corresponds reasonably well to the single-cell sequencing data we have, whereas the targeted assays, they're variable. Some are okay, I'll call them from okay to maybe not so good. That one's not so bad, but the sequencing-based readouts do match up pretty well with your sequencing-based readouts you would get from straight up dissociated cells. This is done with 10x. At the end of the day, you do get, as I say, roughly about 250 transcripts or so per cell with this. It's more than you'll get for the other platforms in terms of total transcripts, although the amount for any given gene is usually lower than for a targeted assay, if that makes sense. So, but nonetheless, you do get more transcripts, and then you do get more genes, of course, because you're covering 23,000 genes versus the targeted assays are going after whatever you've targeted. So the point out of all this, it's very complementary to what a targeted assay do, and it does have discovery capabilities in as well. And then the last thing I'll just mention is that we are doing a lot of proteomics these days with Olink. And so for those of you, I won't get into that, that uses capture probes with paired end oligos, if you will. And at the end, it has a sequencing-based readout. And because the T7 is fairly inexpensive, it's a fairly inexpensive way to get, again, at least cuts that end of the cost. The antibodies are still pretty expensive, but the sequencing cost is now cheaper than the antibodies, which is good. Okay. So overall, what I've told you is that we believe we can get pretty accurate genome sequencing with the T7. You can get these nice phased genomes using LFR, and that technology I showed you was one of the first ones done with this. We think the LFR for the cDNA, for the RNA-seq, is actually quite valuable as well, especially for following allele-specific expression. And then I mentioned the Stereo-seq work that we've done for spatial mapping and transcripts. So as you can see, we're using complete genomics in a variety of fashions. These are the folks who did the work. The RNA analysis on my RNA and genome was done by Jennifer Lee-Bouton with help from others. Single cell, mostly led by Chen-Chen Zhu and others. And Olink, I have a number of people in the lab doing that. And it was all collaboration with Complete Genomics, especially with Brock Peters. I'll give a shout out to him. So I think we're taking questions then at the end of this, and I'll take them then. Thanks.